You should know the female reproductive anatomy that you see in this diagram here on a mid-sagittal section. You will either label that on a diagram or like a photo or something, and the 3D models that we have in lab as well. So again, following the kind of the openings from anterior, posterior, and the female, you have the urethra, the urinary opening into the bladder. You have the reproductive opening, the vagina, and on into that the uterus, and then the digestive system. You have the anus, on into the rectum and the sigmoid colon. Again, this is a mid-sagittal section, but not every structure seen is midline. You have the labium minus and labium majus that you see below. A lot of text you refer to the labia minora or majora. I believe these labium minus and labium majus are singular Latinized terms of that. But regardless of how you know that, the minor and major labia. Again, you also see the clitoris on mid-sagittal section, as well as the pubic symphysis. Again, the clitoris and the penis are both attached to the pubic symphysis and the pubic arch. The other non-midline structure that you see is just above the uterus. There you see the ovary and the ampulla with the fimbriae, the distal end of the uterine tube as well. So external anatomy, you should know as well. Again, the vestibule, the vagina is just the opening between the labia minus or labia minora, and the vestibule includes the vaginal opening and the urethral orifice, or sometimes meatus. The hole at the end of the urethra is often referred to as the meatus. The labia majora, or labium majus, as you see here, meet anteriorly to form the prepuce, just before meeting to form the mons pubis, the fat pad above the pubic bone, covered in pubic hair. But the prepuce of the female is just like the prepuce of the male. It covers the clitoris, whereas the prepuce of the male covers the glans or head of the penis. Again, the labia minora are mucous membrane structures. The labia majora are keratinized stratified squamous epithelia and covered with hair and look more like the skin. The labia minora are more like the lining of the cheek. Again, they're stratified squamous epithelia, but they are mucous membranes and not keratinized stratified squamous epithelia. Again, the deep structures, you have greater vestibular glands, periurethral glands, and vestibular bulbs that are sort of synonymous with some of the glands that you will find in the male as well. And again, the clitoris has more than just the little part that we see. The large crus is basically the equivalent to the corpus spongiosum erectile tissue, corpus cavernosum erectile tissue that you will see in the male structure as well. So, with regards to the development of the ovum and the follicle, just know the stages of development of the ovarian follicle. You have a primordial, primary, secondary, and then a tertiary / mature / Graafian follicle. It's known by many different names. I don't like how they have the tertiary and mature follicles separated here. If anything, I would say the tertiary is an early tertiary, and the one labeled mature is a more of a late tertiary. But really, the tertiary and mature are typically in most textbooks seen as the same stage. Just be able to label this diagram if I gave you something like this, between primordial, primary, secondary, tertiary / mature, and the corpus luteum and corpus albicans is what you have when after ovulation occurs and the follicle cells that are left kind of degenerate. So you should be able to label just about everything you see here, with the exception of what you see on Part B there on the upper upper right, you should be able to label the parts of the uterine tube, the isthmus, ampulla, infundibulum, and the fimbriae. Again, the fimbriae and infundibulum, they kind of have this wave-like movement that induces the ovulated oocyte to enter into the uterine tube. The fallopian tube is an older term for uterine tube. You don't see that much anymore, but you should be aware of that as well. Again, the parts of the uterus you should know, as well as the layers. You have a perimetrium, which is the outermost layer. The middle layer, muscular layer, is the myometrium, and the innermost layer is the endometrium. The endometrium is that layer that builds up and sloughs off monthly during menses, and as well as can become inflamed in a condition known as endometriosis. The cervix is a kind of a structure that protrudes into the vagina a little bit, so that you have these fornices, of singular fornix. You have these fornices that crop up around the cervix. You see here are the lateral fornix, and if you see it on a mid-sagittal section, you'll see the anterior and posterior fornix. But basically, the cervix extends into the vagina a little bit. So there's that little 180-degree turn there you see at the fornix. The external os is the hole, and an os is a hole, or like the end of a meatus, a urinary meatus in a way. But that is what the obstetrician, gynecologist obstetrician will scrape in order to take cells for a pap smear. And again, don't worry so much about the ligaments and things on the right, with the exception of the suspensory ligament. You should know that as the one that carries the ovarian artery and ovarian vein as well. So during the pap smear, the cells, stratified squamous epithelial cells, are scraped off of the os of the cervix and smeared onto a slide and stained. If you remember from staining your cheek cells in 1101 lab, they should look very similar to the cells you see on the right. At the left, under normal cells, with the exception they probably have a blue stain. But on the left, under A, normal cells, you're looking at just a large overlapping cells, very large cells, with a small nucleus within those cells. The cells on the right, you notice the nucleus is very enlarged. The cytoplasmic volume has been reduced, and this is stage 3, what they refer to as cervical intraepithelial neoplasia, which is a fairly serious cancer at this point. So again, with regarding to blood vessels, just be aware that there is an artery for the vagina and the uterus that is a branch off of the internal iliac, and a ovarian artery branches off the abdominal aorta as well to travel through that suspensory ligament to deliver blood to the ovary. Again, just know this stage, the various stages of the follicle development. Again, this is out of a different textbook than the ovary diagram we saw earlier, where you have the tertiary follicle being that stage between secondary and ovulation. So tertiary, mature, Graafian, they are all different terms for the same stage. Again, this is a process, and within that process, there are changes that you can definitely say are this is late stage and this is early stage. So what you'll see is going to be exactly the photos that you see here. Again, photomicrographs of, you have primordial follicles in photograph A there that are they hang out at the cortex of the ovary, and more within the deep to that part of the cortex with the primordial follicles, you'll see primary follicles developing as well. Primary follicles are a little bit larger, and they have a nice row of single simple cuboid cells, follicle cells surrounding them. And again, what you see in B is a mature Graafian follicle with the large antrum, open space, open fluid filled space, and the oocyte with the zona pellucida around it, perched on a little pedestal almost of granulosa cells. So just be able to recognize those again if I ask parts of follicle development within the ovary. So the urogenital triangle is shown here on a male, but it is part of the female anatomy as well. It's just a descriptive reference point that uses the ischial tuberosities, the pubic symphysis, and the coccyx as the points of reference in these two triangles. So just like the female, you should know the reproductive structures of the male mid-sagittal section. And again, obviously not everything that we see here is actually mid-sagittal. The testes are obviously not on the midline, but you'll see the testes, the ductus deferens, the ampulla, seminal vesicle, things like that. Those are kind of off the midline, but the ejaculatory duct is reaching midline. It's something off midline going towards the midline, and where the two ejaculatory ducts meet the urethra in the middle of the prostate, that is a midline structure. So again, just know your different erectile tissues within the penis. You have two corpus cavernosums, one corpus spongiosum that surrounds the urethra. The prepuce, again, you see in the male, slightly different structure than the prepuce of the female, but serves the same purpose, is to cover the glands of the penis or the glands of the clitoris. Again, you see the three parts of the urethra: the prostatic, membranous, and spongy urethra, but you'll actually see them on the next slide. So this slide is the ductwork for the reproductive structures that has basically been dissected out, so you can just see the ductwork by themselves. So sperm are created within the testes, within the seminiferous tubules. You see they're not quite labeled, but you can see the little lobules within that left testes there. They mature within the epididymis, and during ejaculation, the sperm leaves the epididymis, travels through the ductus deferens, sometimes referred to as the vas deferens. This is again an older term. If you had a vasectomy, you had the vas deferens was cut to prevent pregnancies. Again, the ductus deferens leads into what's referred to as the ampulla, a larger swelling of that ductus deferens just before it meets with the fluids of the seminal vesicle at the ejaculatory duct. So as the ejaculatory duct is where the sperm meet the fluid of the seminal vesicle, and those two enter the prostate. The prostate, seminal vesicle, and bulbourethral gland are those three glands of the male that contribute to the volume of semen. So semen consists of the sperm cells themselves, and again fluid from the seminal vesicle, bulbourethral gland, and prostate glands. So again, leaving the prostate, the part of the urethra that travels to the prostate is referred to as the prostatic urethra. You have the membranous urethra / external urethral sphincter that the males have, the females do not, as that urethra passes through the abdominal wall, and as you pass through the penis itself, the urethra passes through what's referred to as the corpus spongiosum, so it's referred to as the spongy or penile urethra, as that urethra leaves through the penis. So cutting the penis reveals the erectile tissue chambers. Again, you have two corpus cavernosums, left and right. You have one singular corpus spongiosum that surrounds the urethra. Within the corpus cavernosum, you have deep arteries, and you also have dorsal arteries and veins on what's referred to as the dorsal side of the penis. So erections basically are kind of again the concept we refer to with glaucoma. You have fluid going in a certain chamber, fluid going out of a certain chamber, and when fluid coming in is greater volume than fluid coming out, the fluid accumulates and starts to build. It builds pressure, and just like the same type of turgor pressure they see operating in plant cells, that's basically a turgor pressure, that pressure of the fluid against the connective tissue of the corpus cavernosum. That rigidness is what actually creates the erection. Same phenomenon happens in the clitoris as well, but it is not quite as evident as the erection seen in a penis, obviously. So again, structures of the penis that you see here that I think are important to know. Um, I think that the three things we really talked about are the cremaster muscle, the dartos fascia, and the pampiniform plexus. The dartos and cremaster muscles, those both to help to raise the testes, to get them closer to the body. Again, the testes have to be slightly cooler than the body for proper sperm production. So the testes are held outside the body, and temperature regulation takes place by contracting or relaxing muscles to bring the testes closer to the body for warmth, or let them hang farther from the body to cool them off. The pampiniform plexus we will see more on the next slide. So the pampiniform plexus acts as a countercurrent heat exchanger. You have warm blood flowing in one direction and cool blood flowing in the opposite direction, just adjacent to the warm blood. So you have a heat transfer from the warm artery descending from the abdomen to the testes. At every point of contact between that artery and the vein returning cooler blood from the testes, at every point in contact there's going to be a gradient of temperature. So temperature or heat is continuously being transferred from the artery to the vein, and basically the effect of that is that as the blood descends into the testes, it can become cooler than it would if those two blood flows were in the same direction. Part of the key of the countercurrent heat exchanger is the countercurrent part, where you have two flows occurring in opposite direction. If the two flows occurred in the same direction, eventually there would be enough heat transferred to where there would be an equilibrium reached, and there would no longer be heat transfer. But because these two flows are in opposite directions, there is a continuous flow of or transfer of heat all the way through this pair of arteries. So again, know the structure of the testes. The testis itself is surrounded by a covering called the tunica albuginea, which kind of turns into septa, little walls, and divides the interior of the testes into little lobules. Those lobules are filled with coiled seminiferous tubules. Those seminiferous tubules are where the sperm are actually produced. So the sperm cells are produced within the seminiferous tubules of the testes. They travel through the reedy testis and kind of hang out in a very large supercoiled tube called the epididymis. The epididymis is where the sperm mature and become motile, and during ejaculation the sperm will leave the epididymis and travel through the ductus deferens up into eventually into the ejaculatory duct and into the prostate. So as you notice, leaving each testis, you have a spermatic cord that is part of that part of the tunica vaginalis layer, the outermost layer of the testes. The spermatic cord has four structures that are important: the ductus deferens, an artery, a vein, and a nerve. Okay, so those are the four things that travel through the spermatic cord. You should probably know those as well. So with the sperm, just know the structure of the mature sperm. Just know the head, midpiece, and tail, and just know a little bit of what's located with each. The head contains the nucleus, which is the genetic material, half the genetic material from that male. The other thing in the head is the acrosome, which is a kind of referred to as a cap. It is enzymes that help the sperm to digest away the cells from the zona pellucida to get through the kind of protective layer of the oocyte. The mid piece is rich in mitochondria that provide energy for the tail, and again the tail is what actually propels the sperm and moves it along through the female's reproductive tract.